So good evening everyone. So today we are going to discuss for the week 11 of medicinal chemistry, which is a course running on NPTEL platform, and the course instructor is Professor Harina Chakrapani from ISET Pune, and I am Simran, your PMRF teaching assistant from IIT Bombay. So till now we have taken 10 classes, 10 tutorial classes of this course, and this is the 11th one. This is the week 11 of this tutorial class. So before going to start, this is some standing instructions that this is a problem solving session which is on every Saturday 5 to 7 p.m. and the platform is Google Meet. In case of any disruption, please wait and do not leave the lecture. And please ask your queries as much as possible in the chat box or raise your hand during the lecture. Slides will be shared with you guys soon, and if you want to see any of the video which we have covered so far in the 10 lectures. So I've uploaded that on this channel, that is in NPTEL-TA. This is my YouTube channel. So I have uploaded all the videos, whatever I have taken so far on this channel, and you can go through that if you are unable to attend any of the lecture. And please make sure to attend every tutorial so to understand the course better, and it is also important for you to have some clarity what I'm talking about. So these are some instructions. So previously I have talked about drug receptor targets, those things we have talked, and here last week we have talked about the prodrugs and interactions related to receptor and target molecule. So based on that, today also we are going to talk about those things and see some of the examples and some of the questions related to that. So let's get started with tutorial questions. So first question is, which of the following is not a characteristic of a good growth drug? Predictable conversion to the active drug, non-toxic metabolites, increased stability in the gastrointestinal tract, immediate activity after administration. So you have to answer which of the following is not characteristic of good prodrug. So any guesses if you have taken the lecture of prodrug? What are prodrugs? What are the characteristics of prodrug? Any guesses, any answers related to this? D. So yes, the correct answer is D, that prodrugs requires activation, so they do not act immediately. So a prodrug basically is a biologically inactive compound which becomes active when it enters in your body, right? So it requires some activation, and previously in the previous class we have discussed how enzymes are responsible for activating these prodrugs, like cytochrome P450. That enzyme activate the prodrug in liver. So those kind of thing we have already discussed. So the characteristic is like option A is saying that predictable conversion to active drug. Yes, that is a characteristic of a prodrug. It can convert into the active drug because it is biologically inactive when you are taking it. Okay. Next is non-toxic metabolites. So yes, it is also a characteristic of a good prodrug that it doesn't contain any toxic metabolite. It should be non-toxic containing metabolites which are present in a prodrug. Increase stability in the gastrointestinal tract. So This is also desirable when you're talking about a product, that it should be stable when it is entering in gastrointestinal tract. It doesn't get metabolized completely. It should be, some of the amount of drug should be present to act at the active site of the enzyme, right? So when you are talking about D, that is immediate activity after administration. That is not a characteristic of a drug. Why? Because this requires some amount of activation. This requires activation by enzymatic activity. They do not like get the immediate effect after the administration. So they will not act immediately, right? So this is not the characteristic of a good product. Otherwise, A to C, these are the desirable characteristics of road. So I hope it is clear to all of you. If anyone has any doubt regarding this, okay. So moving forward, so which class of prodrugs requires enzymatic activation? Type 1, type 2, type 3 prodrugs, or none of the above. So prodrugs are classified into classes actually. So these are some type of prodrugs that are present: type 1, type 2, type 3. So three type of prodrugs are available. So in which class do you think it requires the enzymatic activation? Basically, which class of prodrug requires activation through enzymes in which of the following? So, is it the type 1, is it type 2, or type 3, or none of the above? So, what do you think is the class of prodrug which requires the enzymatic activation? Any guesses? Sorry? So, can you repeat? I didn't get what you are saying. Okay. Okay. Right. Right. So, the right answer is type 1 prodrug. So, this type 1 prodrug requires the enzymatic action at the active site. So B and C, that is type 2 and type 3 prodrugs, these prodrugs are typically activated outside the target site. And some prodrugs requires enzymatic action, right? So activation is required for type 1 prodrugs, not to type 2 and type 3. So they are typically activated outside the target site, not the uh, basically at the site of action it doesn't get activated, but outside the site of action, outside the target site, it is getting activated. So those are the type 2 and type 3, right? So that is why only type 1 is required activation through enzymatic activation. Okay, so I hope it is clear to all of you. So now the prodrugs, we have already talked about it, like a decent amount we have given to it. So now there is a class known as this CRISPR-Cas9. So we will talk about this. So what is this? In the late 1980s and 1990s, researchers include Francisco Mojica noticed unusual repetitive DNA sequences in prokaryotic genomes. So these sequences were short, palindromic, and repeated many times with spacer sequences between the repeats. These unique sequences were called clustered regularly interspaced short palindromic repeats. So this is known as CRISPR. CRISPR is the clustered regularly interspaced short palindromic repeats. So these are the so what is this? So these are the sequences which was short, and the forward and the backward is same. When you are reading it, it is the forward and backward is the same when you are reading these things, and repeated many times with a spacer sequence. So this is the spacer sequence, and this is the palindromic segments which are transcribed after some space. So these unique sequences are known as CRISPR sequences. So the function of these sequences was it remains a mystery in early 2000s, but when these scientists like Alexander Biotin and colleagues identified that this protein called Cas9 and proposed its role as DNA cutting enzyme in the bacterial immune system. So basically, this CRISPR sequence is nothing but you have this palindromic segment, then there is a spacer in between, and there is a repeat between the segments. So there is a repeat segment which is present in between a spacer. So this is the unique sequence which is known as CRISPR sequences. Okay. So there is a protein which is associated with these kind of sequences known as Cas9. So Cas9 is a protein that is associated with it, and that earlier it was proposed that the Cas9 has a role in DNA cutting. So it is a DNA cutting enzyme in bacterial immune system. It is present in a microbial genome. So in 2007, Philippe Horvath and his team demonstrated that these sequences work with Cas proteins to provide adaptive immunity against viruses. So when they are working together, this CRISPR sequences and Cas9 protein, they provide the immunity against the viruses. So that is how bacteria get a immunity against viruses. So when you are talking about the sequence and the Cas enzyme, they work together to incorporate snippets of viral DNA into bacterial genome. This allows bacteria to recognize and defend against future infection in three phases. So basically, this is allowing the bacteria to defend against the infections in three phases. How the three phases are done, so we will be looking at these things step by step. So first of all, you need to know that there are CRISPR sequences and Cas9 protein is associated with it, and they are actually giving the bacteria they are giving the bacteria immunity against the viruses. So how now the bacteria is defending against the infections, that is done in three steps, so which are elaborated here. So first step is cutting and capture. So when bacteria are infected by a virus, they use their CRISPR system to cut up the invading viral DNA and insert pieces of it into their own genome as a memory of infection. So what is happening here? So first of all, when you have this bacteria cell and virus is attacking it, so what happens? This acquires spacer DNA from viral infection. So what happens when bacteria is infected? They use this CRISPR sequences. So CRISPR sequences have spaces in between, right? So this CRISPR system to cut up the invading viral DNA. So whatever the viral DNA was, whatever DNA is that virus is having, the viral DNA. What happens? They cut that invading viral DNA, and that viral DNA they insert in between the space. So if there is a space in between these two segments of CRISPR sequences, now they cut the viral DNA and they fix it here at the spacer. Why they do it? In order to get a memory of of that infection. So this is how they get that uh, if some other virus of that kind is invading, that is how they are getting this is the memory which was there, and now if another virus is attacking, they will activate the system like that. So there is a memory of infection which is incorporated into their own genome now. They have a uh, like fighting memory of that infection. So now the second point is monitoring. So basically, bacteria transcribe the spacer in DNA into the CRISPR RNA. So now this is the genome which having the viral DNA of that bacteria. And now what happened that add spacer to genomic CRISPR region. Now they will add a here, and bacteria transcribe the spacer DNA. So they will transcribe this spacer DNA into the CRISPR RNA. So they are transcribing that. So we have already learned about the transcription, translation of DNAs and RNAs. and we are talking about, I think, week 3 lecture. So if you have any doubt in that, you can go through that video lecture of week 3, I think. So you'll find it there. And now what happens is Cas9 and the crRNA form a complex and monitor the cell for any DNA sequence complementary to the RNA. So now what happens? There is a spacer in which this viral DNA will get inserted, and now this DNA will get transcribed into CRISPR RNA. Third one is the defense. So what is meant by this? So here this is the transcription which is happening. First of all, this transcribed genome CRISPR region. This is the transcription which is happening, and it is processed like a CRISPR RNA. So you will get a CRISPR RNA after transcribing the uh, DNA which contains the viral infection DNA as well. So, if matching viral DNA is encountered, the cRNA and RNA Cas9 complex binds to and cuts the viral DNA to prevent it from replicating. This halts the viral infection. So, what happens here? The third one is that if now, just a second. So here what happens in the third step, that defense will happen. So here, if any time they will find a matching DNA, matching viral DNA, now what happens, the bacteria and this cRNA or RNase Cas9 now complex with each other, and it will cut down the sequence wherever they will find the viral DNA, and they stop them from replicating. So this halts the viral infection. So what happens, first of all, in this three phases? In first phase, they will cut and capture the viral DNA from the virus, and then it invade into the this spacer memory. So here, in between the spacer, this they invade this viral DNA segment, and then they the bacteria transcribe the spacer DNA into the crRNA, that is CRISPR RNA. So there is a processed RNA which is forming, and after the formation of that RNA, what happens, whenever they will find any matching viral DNA which is matching with that memory, then cRNA and RNA Cas9, they complex with each other. Here, there's a complexation which is shown. They complex with each other and they cut down the viral DNA to prevent it from replicating. So this is how they halt the viral infection. They will leave the DNA wherever this viral DNA memory is stored. Okay, so that is how they store the memory of viral infection by some virus. So, using this system, bacteria can collect sequences from many different infecting viruses to create a library. Right. So, this is how they will do from for number of viruses. They will do same practice, same three steps, same thing they will do again and again to keep the memory of many viruses, and they create a library over it. So, since the CRISPR sequence is contained in genomic DNA, it is passed on to each generation, because you are now processing that viral DNA into your rRNA, because it is contained in the genomic DNA. That is how they passed that particular information onto the generations. So this is how they inherit the DNA memory of that particular virus, how to fight them, how to do that. They pass it on to the each generation. And the library continues to change and adapt to more common threats over time. So over time, they will again adapt and change their memory because of some of the viruses which are now maybe adaptable to those conditions, and that, like that. So this is how bacteria fights against the viruses, by keeping a memory, by keeping these three steps. This is how they will incorporate in their genome DNA that how the viral DNA looks, and how they have to cut their DNA sequence whenever they found some infection or viral DNA. So this information they pass on to each of the generation. So again, I have some of the slide which is incorporating this thing. So if I hope this much is clear to all of you, then I'll go forward and we'll talk about in more detail about these four steps. So if anyone has any doubt regarding this cutting, capture, monitoring, and defense of the bacterial DNA, so uh, let me know. Otherwise, I'll move forward with this five-step elaborative way. Anyone has any doubt regarding this particular thing? Okay, so if not, then I'll move forward. So first of all, cutting the DNA. So as we have discussed already, Cas9 enzyme, an endonuclease that cuts both strand of DNA at a specific site. So this is a particular enzyme which is responsible for cutting of the DNA at specific sites, right? So this multiple type of Cas enzymes are found in nature, but Cas9 is commonly used in lab. So single guide RNA, that is known as sgRNA, an engineered RNA that forms a complex with Cas9, and this sgRNA is a fusion of two regions that occur as separate RNAs in nature. So basically, this is some of the summary of enzyme. Um, you don't have to you just have to know that how this CRISPR RNA or cRNA and sgRNA is made. So part of the CRISPR RNA in nature, a 20 nucleotide region that is complementary to the target region and defines the target DNA sequence that Cas9 cuts. So basically here, first of all, Cas9 binds an sgRNA. So this is how it works. Cas9 recognizes and binds the scaffold region of sgRNA, and the nucleotide sequence of the scaffold region determines its structure, which is tailored to fit within the Cas9 protein as a key fits in a lock. So this is how a key fits into the lock. Cas9 binds with sgRNA, that is known as single guide RNA. So here this is something similar to what we have discussed previously. So just it is in the elaborative way. So I'm just reading it out, and you can have these slides and you can go through these things. It is similar to what we have discussed. So step two is the repairing the break to engineer the change. So researchers can use the cell's own DNA repair machinery to modify, insert, to delete a nucleotide sequences. The repair can happen in two ways. So first way is non-homologous end joining, that is known as NHEG. EJ. So enzymes reconnect the ends of the double-stranded break back together, and this process may randomly insert or delete one of the bases and can cause mutations that can disrupt gene function or expression. So basically, Cas9 requires a particular palm sequence to be present directly adjacent to the photospacer where the Cas9 and sgRNA complex recognizes and binds up. So you don't have to go into that deep. You just have to understand how this bacterial cells are fighting against the viral infections and how they have done it. Okay. So I hope this much is clear to all of you. So coming to this question, next question, which is which of the following diagram has the correct numbering system for fluoroquinolones? So this fluoroquinolones we have already discussed in I think week 6 or 7 class. So they have some of the properties. So here you have to answer that which of the following diagram has the correct numbering system for fluoroquinolones. This is the numbering which is done in the fluoroquinolones. So you have to identify which of them is the correct numbering system. Is it the diagram A, B, C, or D? Does anyone knows the answer? So what do you think is the correct sequence numbering of that fluoroquinolone? Okay, so first of all, we'll see how the fluoroquinolone nucleus looks. So this is the standard numbering for the quinolone or fluoroquinolone nucleus. It is a bicyclic ring system, right? That follows the IUPAC rules, which ensure the heterocyclic atoms and key substituents are given the lowest possible lookings. So see how it is going to be numbered. So first of all, you have to start at the nitrogen atom because it is the halogen atom. So according to IUPAC, these are to be numbered as the lowest possible. So n will require at the position 1. So if you are numbering from here, you will get that n is at one position, and then you will move clockwise so that you will get a lowest number for this carbonyl groups. So this will get 3, this will get 4, and the fluorine substituent usually at position 6 is correctly placed. So when you are talking about this, you will get this 1, 2, 3, 4, 5, 6, 7, and 8. So, according to IUPAC rules, you number heterocyclic atoms as the lowest possible. And also, when you are talking about the functional groups, also you want that it will come at the lowest. So, to considering that thing in mind, so you have to number this fluorophore. So, the position goes like 1 is nitrogen in the cumulum ring, that is this. Then this carbon adjacent to the nitrogen, then this carbon bearing a carboxylic acid group, then carbon bearing a ketone group, and the carbon in the benzene ring continues like clockwise. So this is how you number the fluoroquinolones. So this is basically a IUPAC question. It is not related to what medicinal chemistry is. It is just to get you an idea how you are going to number, how you are going to number these kind of molecules, that is fluoroquinolones. So if it is the case, so now what is the right answer in between these four? So it will match with diagram A. Okay. So A will be the right answer. So here I have taken this naming and nomenclature of this fluoroquinolones. This one we have discussed previously as well, but here if you are not aware of these things, we can discuss it here also. So fluoroquinolones are typically named as the suffix fluoraxin, derived from the core quinolone structure with the addition of fluoxetine. So here are example for fluoroquinolones with their nomenclature. So this is norfloxacin and ciprofloxacin, levofloxacin. These are the things which are the fluoroquinolones. And here the mechanism of action is like, so they act by inhibiting enzymes involved in the bacterial DNA replication and transcription. So what they do, they inhibit the enzymes which are involved in the bacterial DNA replication and transcription. So it doesn't allow bacterial DNA replication and transcription to happen. So specifically, they target DNA gyrase and topoisomerase 4, which is responsible for DNA replication and transcription in bacterial DNA. So which are essential for the process of DNA supercoiling and segregation during cell division. So these enzymes are distinct from the ones in eukaryotic cell, making fluoroquinolone selective for bacteria. So the mechanism of action you need to know is that it inhibits enzymes involved in the bacterial DNA replication and transcription. So if we are targeting this DNA gyrase and topoisomerase, so first of all you need to know what are these DNA gyrase and topoisomerase for. So this is what we are talking about bacterial cell. Okay, this is all about the bacterial cell. So DNA gyrase, this enzyme is primarily involved in negative supercoiling of DNA, which helps to relieve the torsional strain generated ahead of replication fork. So this thing of supercoiling that we have discussed in week 2, I think. So there I have taken transcription, translation of DNA and supercoiling. So there are basically three structures of DNA: primary, secondary, tertiary and quaternary. So in quaternary structure, you will see that there is a supercoiling of DNA which is happening to stabilize the DNA. So this DNA gyrase is actually involved in those supercoiling of DNA to release the torsional strain. So this supercoiling is necessary for proper DNA replication and transcription. So DNA gyrase guide that. Okay. So topoisomerase is important for the segregation during the cell division. So when the cell is getting divided, the bacterial cell, so here the segregation is important. That is done by topoisomerase 4. So this is what the mechanism of action of this fluoroquinolones. So I hope it is clear to all of you how this fluoroquinolone is numbered and how it is acting into the bacterial cell. So what they are doing. So if anyone has any doubt uh, till now, so you can ask. If anything is not clear so far, okay, so I assume everything is clear to all of you. So now coming to pharmacokinetics of fluoroquinolones. So this pharmacokinetics and pharmacodynamics, these two terms are very important when you are talking about medicinal chemistry. So these we have discussed in very brief in the previous classes. So here I will take this in not in brief, just taking it as fluoroquinolones pharmacokinetics. So these fluoroquinolones have several key pharmacokinetic properties like adsorption, distribution, metabolism, and excretion. So I have told you that pharmacokinetics is something that what is happening to the drug into your body. So basically what your body is doing with the drug molecule. That is known as pharmacokinetics. And when you are talking about pharmacodynamics, that is what the drug is doing into your body. So how it is acting into your body. That is known as pharmacodynamics. Okay, so this pharmacokinetics in fluoroquinolones is, so there are four steps of pharmacokinetics, that is absorption, distribution, metabolism, and excretion. So here, what is meant by this? Absorption is most fluoroquinolones are well absorbed after oral administration, though absorption can be affected by the presence of diamine and cation. So first of all, first step is that your drug molecule which you are taking either orally, either as injecting, whatever you are doing, that is first of all get absorbed in your body, right? So fluoroquinolones are well absorbed into the body after oral administration. So next step is the distribution. So now your drug has to distribute, right, uh, within your tissues, within your organs. So distribution is these drugs have good tissue penetration and can reach therapeutic levels in most tissue, including lungs, prostate, and bones. So fluoroquinolones are also have good tissue penetration. Tissue penetration means they have good permeability, so uh, it can easily penetrate into your tissues and penetrate into your organs. Third is metabolism. So metabolism is how your drug is getting metabolized in your system. So they are primarily metabolized in liver, though the specific metabolic pathway varies for each drug. So most of the drugs are getting metabolized in the liver, right? So various drugs have various metabolic pathway, but generally what happens that your primary metabolism is taking place in liver. So now the fourth point is excretion, how the drug is excreted from your body. So how it is going to excrete. So most fluoroquinolones are excreted by kidney, making those adjustments necessary in patients with renal impairment. So fluoroquinolones are mostly excreted by the kidney. So these are the four things which is involved in pharmacokinetics, that is absorption, distribution, metabolism and excretion. So this is how your body is absorbing, distributing, metabolizing and excreting your drug. So this is what your body do to drug, that is known as pharmacokinetics. Next point is resistance mechanism. So bacterial resistance to fluoroquinolones can develop through various mechanisms: point mutations, ilex pumps, altered permeability. So bacterial resistance can happen when you are using fluoroquinolones. See, whenever you are using any medicine, there could be resistance which can develop. If you are taking that medicine regularly, or if you are taking it quite a bit, then it is possible to get a resistance over that medicine, right? So bacterial resistance is also similar to that thing. So it can happen to fluoroquinolones. It can develop. How it is going to develop that? Point mutations in genes encoding DNA guidance or topoisomerase 4 can decrease the binding affinity of fluoroquinolones. So there could be mutation which could happen in the DNA sequence that can make the fluoroquinolones bacterial resistance. Okay, so fluoroquinolone, the bacteria could get resistance to the fluoroquinolone. Second point is efflux pumps. Bacteria may increase the activity of efflux pump that actively expel the drug from the bacterial cell. So if your drug is actively getting expelled from the bacterial cell, then it might get affected, and now the activity of your drug will get drastically decreased. So that is how now bacteria develop a resistance to the fluoroquinolones. Third one is altered permeability. So bacteria may decrease the permeability of their cell membrane to fluoroquinolones. So this is how they uh, mutate themselves. They resist themselves from fluoroquinolones. So these are the resistance mechanisms. So I hope this is clear that how fluoroquinolones are numbered, how they are, how they do the mechanism of action of them, and how the pharmacokinetics and bacterial resistance happens to fluoroquinolones. So this is all about the fluoroquinolones acting into the bacterial cell. So I hope it is clear to all of you. If anyone has any doubt in any of the slide, please let me know. Okay, if not, then I'm moving forward. So the next question is, what structure or structures are formed if the acyl side chain of penicillin is hydrolyzed? So they are asking, if the acyl side chain of penicillin got hydrolyzed, what do you think would be the structure? Is it penicillinic acid? Is it penylic acid? Is it 7 amino penicillinic acid? Is it 6 amino penicillinic acid? So we have also learned about the penicillin, how it is uh, how this drug act and how it is the antibacterial drug. So we have in thorough gone through that mechanism of action of penicillin. So if you recall the structure of penicillin, you can surely answer this question. So any guesses? Just think about it. So first of all, we need to know the penicillin structure. So this is the penicillin G. So the key step here is hydrolysis of the acyl side chain. So the acyl side chain attached to the penicillins, these acyl side chains. Penicillin consists of a beta-lactam ring fused to a thiazolidine ring. So this is the beta-lactam ring which is fused with the thiazolidine ring. This is the thiazolidine ring and this is the beta-lactam ring. So this is how penicillin looks along with an acyl side chain that varies among different penicillin derivatives. So there are different penicillin derivatives which contain different type of this side chain. This R will vary for different penicillin category. So this side chain, beta lactam ring and this thiazolidine ring. This is the core structure for penicillin. And the question is asking what will happen, what will form if the hydrolysis of acyl side chain happens. So this acyl side chain will get hydrolyzed. What will happen? So the acyl side chain is attached at the position 6 of the beta lactam ring. Hydrolysis of the amide bond between the beta lactam ring and the acyl side chain breaks the bond, effectively removing the side chain. So basically, there is an amide linkage which is present, and now this amide bond will break in order to hydrolyze this acyl side chain. So what will happen after breaking? This is the molecule which you will get. This is the hydrolysis which will happen at this amide linkage. So what happens? Water molecule will attack onto the carbonyl carbon here. So what happened? This carbonyl carbon is being attacked by the water molecule, and then there is a again hydrolysis which has happened between this amide linkage, and this is how you will end up having this 6 amino penicillinic acid. This is known as 6 amino penicillinic acid. So when after the hydrolysis you will end up having this aldehyde and amine. So after hydrolysis you will end up having this 6 amino penicillinic acid. So when the acyl side chain is hydrolyzed, the molecule retains the structure of penicillin core with the amino group here at the position 6 still intact. So rest of the ring will remain same, just the hydrolysis happen at the amide linkage, and here after the hydrolysis you will end up having this aldehyde plus amine. So the hydrolysis does not alter the amino group at position 6. So it simply removes the side chain that was originally attached at position 6. So you doesn't remove this amino group at the C6 position, only this amide linkage will get hydrolyzed. So after the hydrolysis, the remaining structure is 6-APA, that is 6 amino penicillinic acid. So this structure is known as 6 amino penicillinic acid, which consists of penicillin core, that is beta lactam ring and thiazolidine ring. So the penicillin core remains intact with the amino group at position 6 and no side chain at position 6. So there would be no side chain which is present at the position 6, only the amino group will be present after the hydrolysis of penicillin. Right? So if this is the if you are talking about this penicillin G, so what happens here? The hydrolysis has to happen. Now this amide linkage will get disrupt and you will end up having this 6 amino penicillinic acid and phenyl acetic acid. So there could be different kind of acyl side chain in different penicillin categories, but after the hydrolysis, you will end up having this 6 amino penicillinic acid in each of the case, and just the side product will be different in every penicillin category. It's just 6 amino penicillinic acid you will get in each of the following penicillin derivative after the hydrolysis. Okay, so hydrolysis product is 6 amino penicillinic acid. So the right answer is 6 amino penicillinic acid. So I hope it is clear to all of you how your penicillin side chain is getting hydrolyzed to give you 6 amino penicillinic acid. If anyone has any doubt, please ask me. If it is clear, then moving forward. Just- Okay, so you already know the answer. So here the question is the following structure shows the pharmacophore for tetracyclines. Which of the heteroatom indicated could not act as a hydrogen bond acceptor? So the structure shows the pharmacophore for tetracyclines. So this is the tetracyclic MT. This is a pharmacophore compound. You have to answer that which of the heteroatom, which is indicated like A, B, C, and D, which of them could not act as a hydrogen bond acceptor? Is it D? Is it C? Is it B or is it A? So which of the heteroatom you think is not participate in hydrogen bond acceptor? Heteroatom A, B, C or D? It is a very simple question in terms of hydrogen bond acceptor and donor ability which we have discussed. So any answers? Think about it. Okay, so this hydrogen bond acceptor and donor ability. So if you are talking about this, then this heteroatom B that is here, it doesn't act as a hydrogen bond acceptor. Why? Because here, when you talk about an acceptor, so one by one we will take this question. So here, heteroatom A, that is this tertiary amine. So first of all, see this tertiary amine, it has lone pair of nitrogen. Yes, but the nitrogen is sp3 hybridized, and the lone pair is an orbital that can accept hydrogen bonds. But the lone pair is less basic. There is a steric hindrance because of the two dimethyl groups, and also and nitrogen is less electronegative than oxygen. So weaker hydrogen bonding. So they will result in very weak hydrogen bond acceptor, but this could accept hydrogen bond. Okay, it could be weaker, but it could accept because of the lone pair of the nitrogen which is present. And when you are talking about the oxygen, that is this carbonyl. If I take this carbonyl oxygen, this one, so what will happen now? Uh, so first of all, we'll take this carbonyl oxygen. So see, it also have lone pair of electrons, and it is strongly electronegative as well. In theory, a great hydrogen bond acceptor is water, protein, and backbones. So, however, in conjugated system, the lone pairs can be delocalized into the pi system. So here, as you can see here, that there is a conjugated system which is present. This is the beta alpha beta unsaturated ketone. So in conjugated system, the lone pair can be delocalized, and this reduces their availability to accept hydrogen bonds. So in some cases, carbonyl oxygen is excellent hydrogen acceptor, but in conjugated system, it is less effective. So it is less effective, but it still is a hydrogen bond acceptor. It is stronger than this tertiary amine as a hydrogen bond acceptor. And when you are talking about this amide oxygen over here, this oxygen, so what happens in the amide case? This oxygen is a part of resonance structure. How come? See, this nitrogen lone pair is involved in the delocalization with it, right? So it is in part of conjugation. The lone pair of electrons of nitrogen delocalize into the carbonyl system, and this shifts the electron density and makes the oxygen less electronegative and less available as a hydrogen bond acceptor. So amide oxygen are weaker acceptor than ketone or ether. So till now we have seen that they are weak acceptors, but they are acceptors, right? So tertiary amine is very weak. Then we talk about this amide oxygen, and the third one, which is very strong acceptor, that is carbonyl oxygen. And when we are talking about amine, so this amine, because it has a, it can act as a donor because of the NH bonds. It can act as an acceptor but very weak because the nitrogen has a lone pair. However, in primary amines, the lone pair is not delocalized and there is less steric hindrance. So it can accept hydrogen bond, but it is a better donor than acceptor. It can although accept hydrogen bond, but it is a very good hydrogen bond donor than acceptor. So that is why we have concluded that it do not act as a hydrogen bond acceptor, this amine. Instead, it will act as a hydrogen bond donor. It will donate these hydrogens to another hydrogen bond acceptor molecule. So, if you compare this tertiary amine, carbonyl oxygen, amide oxygen, and amine group, this amine group is better donor than acceptor molecule. So it is a good hydrogen bond donor rather than acceptor. So that is why the correct answer is heteroatomy, that is this primary amine group. So I hope it is clear to all of you. If not, just let me know so that I can explain again. Anyone has any doubts? Okay, so if not, moving forward. So this is a basically comparison window we say. So tertiary amine lone pair availability, yes, but hindered. So all of them has that lone pair availability, but in this there is no delocalization. In amide there is a strong delocalization, and we are talking about this hydrogen bond acceptor quality. When it is weak, it is moderate, weak and moderate. So because it is moderate hydrogen bond acceptor, but amine will go for donation rather than acceptor because it is a very good donor. So that is why it is hydrogen bond donor rather than acceptor. Now coming to the next question. The following structure, cephalexin, is a first generation cephalosporin. So you have to answer it is a good for oral activity, it is a good living group, it acts as a steric shield, it is generally good for activity. So this is the structure of this cephalexin, which is a first generation cephalosporin. So you have to answer which of the following characteristic resembles this structure. Okay, I think this is a bit difficult. So, first of all, we will understand what is this cephalexin. So, this cephalexin molecule has a primary amine with it in the C7 side chain, specifically a phenylglycine motif. So, this is a phenylglycine moiety which is present and due to which it contains a primary amine. This plays a critical role in oral bioavailability. So why this particular thing is playing a good role in the bioavailability? Because it is acid stabilized. How is it acid stabilized? The amine group which helps stabilizing the beta lactam ring in the acidic environment of the stomach. So this amine group which is present here, it actually helps in stabilizing what this beta lactam ring. This beta lactam ring which is present here, it helps in stabilizing that in acidic environment. This prevents the degradation of the drug before it can be absorbed, unlike many cephalosporins which are destroyed in the acid. So see, there are various generation of this cephalosporin. In this case, this amine group make this molecule tolerant in the acidic condition. So how, if the acidic condition is there, instead of this beta lactam ring opening, what happens first? This nitrogen lone pair will attack onto the acid to form this ammonia, right? So the acid first react here rather than reacting here. So it prevents the degradation of the drug. It makes the drug stable in the acidic condition so that it can be absorbed. Second is enhanced absorption. So the absorption is now enhancing because of this amine group. How does deuterionic nature, presence of both positive and negative charges, help with passage through the gut membrane? So now you will see that in the acidic and basic condition, what happens? There is a anionic charge and positive charge, which will which can create here. So there is a bitter ionic nature. This compound has a bitter ionic nature, means it can have presence of both positive and negative charges. So presence of this both positive and negative charges help this particular compound to pass through the gut membrane. So this amine group can interact with transporters, improving intestinal absorption. It can act as a handle. So this handle can react with the transporters which is present in the intestinal moiety, which improves the intestinal absorption. So cephalexin is orally bioavailable. That's why it is the one of the few cephalosporins given orally. So because it is orally bioavailable, because of the stability of that molecule and absorption, which is very good, because of the presence of amine group and the zwitterionic nature, this is generally given orally. Okay, so it is given orally, that is having a good oral activity. So the in between these four, the correct answer is having a good oral activity because of the presence of this amine group, which improves the stability of the molecule in the acidic condition. Basically, acidic condition is in our stomach, so it will stabilize that particular drug into our stomach. It doesn't get metabolized. Then after the stability, it comes that absorption is very good because of the deuterionic nature. So that is why it is a good medicine to be given oral. It doesn't act as a living group. It acts as steric shield. No. It is generally good for activity. No. So the main reason is it is good for oral activity. So this is how the cephalexin is given orally and have good oral bioactivity due to the presence of this amine and zwitterionic nature of the following compound. So I hope it is clear to all of you. Now we'll see why other options are incorrect. So first of all, just tell me that this part is clear to all of you. Any doubts in this part? Cephalexin. How this primary amine side chain is improving the stability and absorption of that compound. Any doubt in this? we'll see why other options are incorrect. It is a good leaving group. Leaving groups are relevant to the reactivity of beta-lactam with BP, that is penicillin binding proteins. Amino group here is not a leaving group. In fact, it is retained during the action. So that is why it is not a good leaving group, because amino group is not going to leave the molecule. It is going to retain in the acidic region also. Second one is it acts as a steric shield. Steric shield prevent enzymatic degradation. However, this amine group does not provide bulky protection. So steric shield basically works if your nitrogen will have dimethyl, then it will give you a steric shield and prevent the enzymatic degradation. So that is how they improve the stability of that molecule. It doesn't get metabolites, it doesn't get degraded by the enzyme uh, very soon in the body. However, amine group, as it is not a bulky compound, it is not a bulky protection. So it doesn't act as a steric shield here. Other cephalosporins use methoxy or other groups for that. So there are some modification of this compound which uses that thing, that is the methoxy groups or dimethylamine groups to do that. That is steric shield. That is known as steric shield. Third one is it is generally good for activity. So while the side chain influences activity, the term generally good is two-way. Its main contribution is to oral activity, not necessarily increase the potency across all bacterial strains. So this amino group is good for activity. You cannot say that because the main contribution of this amino group is to increase the oral activity. So basically to increase the oral bioactivity by absorbing and getting stabilized in the acidic condition. This is how they are giving that particular thing a oral bioavailability which is increased. Okay, so that is why these three things are incorrect, and only this option, that is primary amine, is responsible for that oral bioavailability, that is good for oral activity. It is the right answer in case of cephalexin. So I hope it is clear to all of you what I am trying to convey. Okay, so if it is clear to all of you, then I am moving forward. So a pharmaceutical lab wishes to synthesize all the possible dipeptides containing the amino acids threonine, uh, phen- uh, phenylglycine, tyrosine, threonine. How would you carry out these synthesis using combinational techniques? So now you have to answer if a pharmaceutical lab wants to synthesize all the possible dipeptides containing the amino acid, these five amino acids. How would you carry out these synthesis using a combinatorial technique? So this combinatorial technique and parallel technique, if you recall, we have actually talked about that also in previous lectures. So you can go through, I think, week 9 or week 10. So in that you will find that we have talked about this combinatorial technique. So the first option is mix all five amino acids together and add the mixture to the resin such that the amino acids are attached to the resin, and then add another mixture of five amino acids such that dipeptides are formed on the resin. So to understand this question, please just go through these four options very thoroughly so that we can see what is the combinatorial technique and how it is affecting it. Okay. Second is split the resin between the five vessels. So you are taking resin in five different vessels, and now you are adding a different amino acid to each of the vessels such that the amino acid is attached to the resin, and now you are mixing the resin beads from all the vessels and re-splitting the beads back into the five vessels. So then add a different amino acid to each vessel to form a dipeptide on each resin. It is a bit confusing, I know, but just visualize the thing. Third one is split the resin between five vessels and add a different amino acid to each vessel such that it is attached to the resin, and then add a different amino acid to each vessel to form dipeptide on each resin. Fourth one is split the resin between five vessels and add a different amino acid to each vessel such that it is attached to resin, and then you have to add a mixture of five amino acids to each of the vessels to form dipeptides. So these are the four techniques of how you can synthesize the possible dipeptides amino acid containing these four amino acids. So how now you are going to synthesize all the possible dipeptides containing these kind of amino acids? Now you'll see. So the right answer is split the resin between the five vessels, add different amino acid to each vessel such that amino acid is attached to the resin. Mix the resin beads from all the vessel and then resplit the beads back into the five vessel, and then add a different amino acid to each vessel to form the dipeptide. So to synthesize all the possible dipeptides using the amino acids, so possible dipeptides which could form if you are having these five amino acids, so it could be 25 combination. So 25 combination is possible if you are combining dipeptide and using five amino acid that is illustrated here. So the this option uses the split and mix technique. So if you want to synthesize all the possible dipeptides in lab, you need to have a split and mix technique. This is a core method in combinatorial chemistry. So how this works? So basically this allows for the systemic and efficient generation of all possible combinations. In this case, all 25 dipeptides. So how it works? How it works? First of all, you have to start with resin beads as your solid support. So what are you doing? You are just adding resin beads in all five vessels. So split the resin beads into the five vessels. So you have splitted the resin beads as a solid support in five vessels, and then you have add one amino acid to each vessel. So in five vessels, I am adding one of the amino acid. So now each vessels have resin with one type of amino acid attached. Right. So first position is occupied by the one amino acid you have already added into the resin. So each of the vessels have one type of amino acid attached in the first position. Right. This much is clear. Now mix all resin bead together. So if you are mixing all the resin beads together, thoroughly combining all the different beads, so you have mixed all the sorts of combinations, and then you have resplit the mixed bead into five new vessels. Now you will take new vessel, that is new five vessel, and now you resplit the mixed beads. Now in each new vessel, you need to add different amino acid again. So the second amino acid coupled to the first one and form a dipeptide on the resin. So now the second position is occupied by another amino group, and this is how you will end up having a dipeptide molecule. So what it will do, this ensures that each of the five first position amino acid is combined with each of the five second position amino acid, and this will give you a result of 25 combination. So this is how you use combinatorial chemistry in order to synthesize dipeptides in of desired combination in lab. So this is a split and mix technique which we will see. Okay. So now coming why other options are incorrect, because in first you have mixed all the amino acid together and add the rest. So this gives a random attachment. This doesn't allow you to have all the 25 combination. It will give you a random attachment and no control over the specific combination. So many beads will end up with mixtures and not all 25 combination will form. So if you randomly mix with 5 amino acid, it will not going to give you 25 combination. It will be a random attachment. Third one is no mixing step after initial attachment. So you are doing the same thing in the third thing also, but what is missing is there is no mixing step after the initial attachment. So if you are doing without mixing the resin beads after the first attachment, you won't get the combination across all amino acid. Only a limited subset will there. So if you read out it, it is the split the resin between five vessels, add a different amino acid to each vessel. That is right, such that it is attached to the resin. This much is right. Then add a different amino acid to each vessel. So you are not splitting, you are not mixing the resin beads here. If you are not mixing it, you won't get the combinations of all the possible amino acids. Fourth one, why is it not correct? Because add mixture of amino acid in second step. So if you see, split the resin between the five vessel and add different amino acid, then add a mixture of amino acid. You don't have to add mixture of amino acid. You need to mix the resin beads and then you have to resplit it. So that step is not right. So this creates randomness in the second step, losing control over the specific payload. Again, not all 25 peptides are guaranteed. So if you want to form a dipeptide, and also there is some desired amino acid which you have to incorporate, you need to use a split and mix technique, which is a method in combinatorial chemistry, and this is how you will end up having all the possible combinations. Okay, so I hope it is clear to all of you how these combinatorial techniques are used in lab to create the dipeptides of your interest amino acids. If something is not clear, please ask me. So assuming that everything is clear to all of you, so I'm just stopping it here today. So we'll see you in the next class. I'm just stopping here, and so I'm just stopping here. I hope it is clear to all of you, and we'll continue in the next class. So thank you so much for attending, and we'll meet on the next Friday. I think Friday I have a extra lecture, and Friday Saturday we'll be meeting in the next week. So hope to see you there. Thank you so much for attending.